Hi everyone. In this lab, I'm going to explain the effect of different temperatures on various bacterial species. So here, the goal of this lab is to learn what happens when we take different bacteria and we grow them at different temperatures. And we're basically trying to determine the optimum temperature for each bacteria. And this is important in medicine because it helps us see what temperatures kill bacteria. So this is why we cook and boil food. And it's important in research because if you know the optimum temperature of an organism, it also helps you determine how you are going to grow it in lab. So if you're doing more research on an organism, and we're going to discuss these terms right here, and we will go through the background and then I will tell you how the lab is done. So just to give a background, whenever we grow bacteria, besides nutrients and oxygen, another very important factor that we have to take into consideration is temperature. The reason why temperature is important, if you guys remember from chemistry, temperature controls the rate of various reaction, and something that's very important in reaction are enzymes. And so enzymes speed up chemical reactions, and so temperature controls the rate of these enzymes. That's why temperature is important. Temperature also controls the fluidity of the plasma membrane. So when we look at a bacterial cell, or all cells for that matter, there's a plasma membrane or cell membrane. When we say fluidity of the cell membrane, we mean how leaky or strong that membrane is. And I'll talk about it more with high and low temperatures. And then finally, temperature also, because it controls these things, which is the rate of enzymes and how fluid the plasma membrane is, meaning letting things in and out, it determines growth. So will an organism grow at one temperature or be killed at another temperature? When we look at organisms, all organisms have a temperature range that they grow in. It's not just like one exact temperature. It's a range, and there's an optimum temperature at that range. The optimum temperature means the temperature where that organism grows the most. So if we were to take bacteria and streak it on different agar plates and stick those agar plates at different temperatures, we would see a range of temperatures that the bacteria grow out. And at some plates, it won't even grow out all, but on the plates we'll see that at one temperature that plate would be filled with bacteria, meaning that the bacteria really like that temperature. So that's called the optimal temperature, where we see the fastest and most growth of that organism. Now keep in mind, for bacteria, there's a low, in that minimum and maximum range, there's the low temperature and the high temperature. For every bacteria, its slow temperature means that the enzymes are very slowed down, so reactions are happening very slow, metabolism is very slow, and at low temperatures the membrane is less fluid, meaning that there's less nutrients going in. That's why typically at low temperatures, such as refrigeration, it slows down the growth of many organisms. So for example, if you take, typically what spoils food is bacteria. It could be other organisms, but if we're talking about bacteria specifically, if you have bread out on the counter versus if you have bread in the fridge, and I'm talking about bacteria, not mold, but we can also think about mold, you will notice that bacteria will go bad much slower in the fridge than the bread that's outside. Same thing with milk. So if we keep milk at room temperature, it will go bad in a few hours. But if you keep milk in the refrigerator, it does eventually go bad. So let's say it goes bad in a week, and that's because low temperatures slow down reactions. So things are still happening, but at a very slow rate. Now, high temperatures generally kill organisms depending on the range of the organism. And the reason why high temperatures are bad is because at very high temperatures, proteins denature, meaning that the shape of the protein changes. If we change the shape of a protein, it denatures, the protein stops functioning, and 99.9% of enzymes are proteins. So proteins denature at high temperatures. This is why fevers are bad for us, because our own enzymes start denaturing. And then the membranes of cells, of bacterial cells, and our cells too, at high temperatures, they start getting ruined. So they start becoming very fluid and leaky. So that's why high temperatures are bad. And then the optimal temperature is the perfect temperature that an organism grows at, and we see the most growth. Now, with the effect of temperature, we as scientists can classify organisms based on the temperature that they like. And we use these terms to classify organisms. Organisms that are cold-loving organisms. So if we look at this graph right here, this on the y-axis shows us the rate of growth, and then on the x-axis is temperature in Celsius. And just to keep in mind, 0 degrees Celsius is where water freezes and 100 degrees Celsius is where water boils. That's why we use the Celsius scale in science. So if we look at this, organisms that grow at cold temperatures or cold-loving organisms are called psychrophiles. So these organisms are found in Arctic, Antarctic regions and in the deep, deep ocean where it's very cold. Organisms that like moderate temperatures or middle-loving organisms are called mesophiles. Almost all pathogenic organisms, so human pathogens, the ones that make us sick are mesophiles because their optimum temperature is body temperature, which is 37 degrees Celsius. Now, there's a class of organisms between the cold-loving organisms, which is psychrophiles, and then the middle-loving organisms, which is mesophiles. And this middle class is called psychrotrophs. So these organisms grow in cold to moderate temperatures, and they're typically the ones that cause food spoilage. So they're between the cold-loving organisms and the middle-loving organisms. And keep in mind that most organisms like middle temperatures. Most organisms are classified as mesophiles, so they like body temperature. Organisms that like heat are called thermophiles. Remember, phile means loving and thermo is heat. And these organisms grow at hot temperatures. They're found in hot springs, very hot environments. And the reason that there are organisms that we classify as thermophiles is because these organisms, unlike most other organisms, have special enzymes, and their enzymes are very highly folded, so they don't denature at high temperatures. So that's why thermophiles can exist. And then organisms that grow at very extreme heat, these are organisms that we classify as hyperthermophiles, and most of these fall into the class of archaea. Remember, archaea and bacteria are two different things, and then there are a few bacteria that are hyperthermophiles, and these are very, very hot temperatures. So like bacteria and archaea that grow in deep hydrothermal vents. So these are the classes I want you to know. And again, keep in mind that most organisms, ones that cause disease, pathogens, are mesophiles. Okay, in this lab, the effects of temperature lab, which unfortunately you're not doing, but if we were to envision that in the online world you were doing it, what you were going to do is you were going to take 4 different bacterial species and streak them on agar slants. So there's just these tubes that have agar in them, and then you were going to incubate or put those agar slants at different temperatures. And then when you come in the next day, you were going to pull all those tubes out and look at them and see what temperatures did the organisms grow at. So the bacteria you are going to test were Bacillus licheniformis, which is a soil bacterium, Bacillus stereothermophilus, which is found in hot springs, E. coli, which is found in humans as part of our normal flora, and then Xanthinobacterium lividum, which is also a type of soil bacteria. So you are going to take these bacteria, and for each one, you are going to slant it at 4 different agar slants. Agar is just the food that the bacteria use to grow. And then you are going to incubate or put these agars at 4 different temperatures. You are going to incubate one of them at 5 degrees Celsius, which is refrigerator, 22 degrees Celsius, which is room temperature, 37 degrees Celsius, body temperature, and 55 degrees Celsius, which is hot temperature. And then you are going to come and look at the agar plates and see where the bacteria grew the best. And remember, optimum growth is where we see the most bacterial colonies. So the idea of this lab, besides figuring out the minimum and maximum temperatures and the optimum temperature of each bacteria, is we wanted you, when you came in, on the agar plates where you saw no growth of bacteria, we wanted you to re-incubate those tubes at the temperature that you saw the most growth at. So for example, if I took E. coli bacteria and I stuck it in these 4 different temperatures, and then I came back the second day, I took out all my tubes from the incubator, the fridge, wherever I put it, and I saw, for example, that the 5-degree slant, so the agar slant that was grown in the refrigerator had no growth. I would re-incubate it at the temperature where I saw growth. So if I saw that E. coli grew at 37 degrees, I would take the 5-degree slant and put it at 37 degrees and incubate for another 24 hours. The reason we're doing this is we're trying to determine if the initial or first temperatures are bacteriostatic or bactericidal. These are very important terms in microbiology that I want everyone to know. Bacteriostatic means anything that stops bacteria from growing, so it inhibits bacterial growth. So if I say something is bacteriostatic, it means it inhibits or stops bacterial growth. So if I say that this temperature is bacteriostatic, it means that it stops the bacteria from growing. An example of a temperature that's bacteriostatic is typically cold temperatures are bacteriostatic. So refrigerating or freezing stops bacteria from further growing, so they're bacteriostatic temperature. Another term is bactericidal. Bactericidal means that something kills bacteria. So if I tell you this temperature is bactericidal, it means that this temperature kills bacteria. Examples of temperatures that are bactericidal are high temperatures, so cooking or boiling. Again, bactericidal means something kills bacteria. Bacteriostatic means that it inhibits or stops the growth. So in the results, what you were supposed to see is, for example, for Bacillus licheniformis, this bacterium, you were supposed to see no growth on the 55-degree slant and 5-degree slant. So this bacteria, like E. coli, does not grow at very cold temperatures, 5 degrees Celsius, or hot temperatures, 55 degrees Celsius, but it grew at 22 degrees, which is room temperature, and 37 degrees, which is body temperature. When we look at the agar slants, we saw more colonies on 37 degrees Celsius than 22 degrees Celsius. So for that organism, we would say that the optimum temperature is 37 degrees Celsius. And when we take the tubes that had no growth, for example, for Bacillus licheniformis, that was 5 degrees Celsius and 55, and we re-incubate them at 37 degrees Celsius, which is the optimum temperature, we see that the tube that was originally in the refrigerator, 5 degrees Celsius, and it was incubated at warmer temperatures, we see growth. So we say that 5 degrees Celsius is bacteriostatic. It stopped bacteria from growing. When we take the 55-degree tube and we re-incubate it at the optimum temperature, we saw no growth. So we would say that the initial temperature is bactericidal, meaning it actually killed the bacteria. So it doesn't matter what you do, the bacteria are dead. So please know these terms. And just to go through them, for Bacillus licheniformis, the optimum temperature is 37 degrees Celsius, so I would classify this as a mesophile. For Bacillus stereothermophilus, it only grew at very hot temperatures at 55 degrees Celsius, so we classify this as a thermophile. E. coli is a mesophile, and Genthinobacterium lividum, its optimum and only temperature it grew at is 22 degrees Celsius, so we would classify this as a psychotroph. And what I want you guys to learn from this lab, even though you're not doing it, is I want you to know what these terms mean. So psychrophile, mesophile, thermophile, I want you to know what they mean. Which one is heat-loving organism, cold-loving organism? I want you to know what optimum temperature means and what it means for a temperature to be bacteriostatic or bactericidal. Know which organism was a thermophile and why. How do thermophiles survive and grow in very hot conditions? So I talked about that. So most organisms do not grow in high conditions because it causes their enzymes to denature. For thermophiles, they're able to grow because their enzymes are very highly folded, so they won't denature at these high conditions. Know the difference between bacteriostatic and bactericidal. And that's it for this lab. So just go through and try to learn the key points in this lab.